So let me introduce our speakers. We have first contacted Karlee at suggestion by Dr. Patty Cleary. So Karlee Wendelberger, soon to be Evergreen, is a laboratory technician at Co-Diagnostics. They are located in Salt Lake City, Utah. Karlee earned a Bachelor's of Science in Biochemistry at UW-Milwaukee in 2019, and she did a summer internship with Dr. Yang there, not the same as our Dr. Yang. She worked 6 years at Adventure Rock Climbing Gym before she found employment at Co-Diagnostics at the beginning of the pandemic last year. So she got thrown right into the deep end. Karlee is half in manufacturing and diagnostic tests. I'm sure we're going to hear more about that, including COVID-19 tests. And she's half in R&D, which is working on a mixture of technology and chemistry. She and her fiance have found a recent hobby in 3D printing. Kyle May is a laboratory scientist at Co-Diagnostics. He earned his Bachelor's of Science in Cellular and Molecular Biology with a chemistry minor. This was in California State University in Chico. He then went to Utah State University for his master's in biochemistry. There he was working with Dr. Joni Hebel's lab. There he was studying the functionality of PRMT1 and non-ribosomal peptide synthetases, or NRPSs. Sorry, that one didn't roll off for me. Prior to working in the laboratory setting, Kyle worked in various restaurants while in school before starting at Co-Diagnostics in May of 2018. Right, so you can get experience anywhere. He is a lifetime snowboarder, but is currently learning how to ski from his wife. I hope she's a good teacher. Jointly, Kyle and Karlee and a few of their lab techs play Dungeons and Dragons once a week, in which Karlee has been 3D printing some pieces for the game. So that's the introduction for our speakers. At this point, I'll turn it over to Kyle and Karlee. Go ahead and take it away.
All right, thank you, Deidre. So a unique RT-qPCR approach to detecting COVID-19 and other infectious diseases. We are working at Co-Diagnostics in Salt Lake City. I'm Karlee Wendelberger, and I'm with Kyle May here. Okay, there we go. So I'm Karlee Wendelberger. I'm a laboratory technician. I started off here as manufacturing mainly, and then I started doing some R&D, and now I'm mostly doing R&D work here. With manufacturing, my main focus was making COVID-19 tests. I developed a protocol to do large-scale manufacturing. We never had to produce liters worth of product at a time. So it was a big change for us. We had to implement a lot more cooling steps to prevent the product from degrading. And now we have a really good system, and we are producing up to like 100,000 tests a week.
Oh yeah, a million.
Or million, yeah, a lot. So I got my biochemistry degree from UW-Milwaukee. I took one extra bio lab course, and they gave me a minor, which was really convenient. A few of my hobbies include 3D printing, indoor hydroponics, mountain biking, and rock climbing. I posted a few pictures with a few 3D prints that I've done. There's a website called Thingiverse, so you can find pre-made designs on there. So I found this amazing Millennium Falcon multi-tube holder, and that was one of the first practical prints that I made for the lab. Since then, I started doing original designs. This gold stand, that's actually a plate for PCR plating. You can put your PCR plate into the stand and then slide it into an ice block, and it'll lock it in place. And it makes it really easy to then pipette into the wells and not have the plate slide around. We've actually had a couple plates drop from ice blocks, so this prevents that from happening. And then I have my D&D character up in the corner. That is my old man, who's a dwarf. One of my coworkers actually printed him on a resin printer, so it's nice and small details. His name's Eppendorf. It's a nice lab joke. Yeah, so I've been doing lots of 3D printing. I just got my first mountain bike a few weeks ago, and I've been a long time rock climber as well. Here's a photo of my fiance and I, that's Lowell. A little bit about my career story. I started high school at Kettle Moraine, and I graduated there in 3 years, and I had above like a 4.0 GPA. I went really hard because I just wanted to get out of high school. I started right away at Florida Southern College, so 17 as a freshman, and I started as a biology major. I did that for a while, and I decided I wanted more math, so I actually switched to biochemistry at the same time when I transferred to UW-Milwaukee. And that's where I did my undergraduate research with Dr. Yang. We focused on type 3 secretion system. I was growing E. coli and then mutating it and observing the gene expression. And that was fun. We can talk more about that at the end if you have questions. But my mentor was Alale, and she was amazing. I would not be where I am today if it wasn't for the undergraduate research that I did. After the research, I moved to Utah, and I found the job listing for Co-Diagnostics on Indeed, and I just applied thinking it was a really cool company. They have a lot of neat patented technology, so I wanted to see what it would be like, and I was surprised that they gave me an interview right away, and they asked me all these goofy questions during the interview and hired me, which I was so excited about. And now I've been here for a little over a year, and it's been an adventure. My first job, like my first real research job right out of college, and having it be manufacturing COVID-19 tests during a pandemic has been really intense. Some of my career goals would be getting my PhD in biochemistry. I really would like to do more independent research in the future. I really think it'd be interesting to do some sort of biochemistry with like astronauts, or somehow work with SpaceX. That would be dream come true. We'll see.
So I'm Kyle May. I am a laboratory scientist at Co-Diagnostics, currently the manufacturing team lead. I grew up in California, about an hour north of San Francisco. I got my BS in cellular molecular biology with a chemistry minor at CSU Chico. After that, I went to Utah State University, where I got my master's in biochemistry in Dr. Joni Hevel's lab. Some hobbies of mine: I've snowboarded my whole life. Last Christmas, I was gifted a pair of skis. So this year, my wife is teaching me how to ski. And so far I have one season under my belt of skiing only. So hopefully next year, I'll actually be able to do something fun. I really like playing video games, board games, card games, any kind of games, really. I play soccer. Couple of years ago now, I started a sourdough starter culture of my own. So I've been learning how to bake some good sourdough bread for the last couple of years. So that's been fun. And you can see that little guy I'm holding. His name is Sensei, and my wife and I just got him over last summer. So a new hobby is playing with our dog. My sort of career story alluded to it a little bit. I grew up in Windsor, California, Windsor High School. I went on to CSU Chico, where I had a business major for the first year, realized I wasn't into it. But I was always really into like the science classes that you had to take initially, and those were always really interesting to me. And so I decided kind of randomly to go with a pre-med focus. And during that time, I sort of thought, I like science, it seems like a worthwhile pursuit to kind of, be able to help people. So I thought pre-med was a great mix of that. And as I was going through that experience, I realized that enjoying science and being able to help people doesn't mean you have to be a doctor. So I started gravitating more towards the research side of like biomedical research type of thing. So I ended up with a cellular and molecular biology major, chemistry minor, and that led me to Utah State after doing some research with some of the professors at Chico who had went to Utah State as well. They told me about Utah State. It sounded really awesome. Utah gets a lot of snow, so that was kind of my pull towards Utah. I ended up getting my master's in biochemistry there, researching these two different protein systems. I met my wife Madeline there, and that's her and I there in that picture. And after graduating from Utah State, one of the people that worked in the lab that I was in, his name was Chad Appley, and he started working at Co-Diagnostics when he graduated. And then he knew that I was graduating the next year, and at the time he was the laboratory director at Co-Diagnostics, and he let me know that they were hiring. So I applied there. I got that job after my graduation, so May of 2018. At the time, I was one of 2 laboratory technicians at the company. And now at this point, we've grown to over 10 different lab techs or lab scientists, and we're looking to be hiring more. So one of the slides at the end of this presentation will be about recruitment opportunities. So if anyone is looking for opportunities at the end, give us a shout. But yeah, so the company has went from 2 laboratory personnel to, growing up to 15 or 20 in the near future.
So we're Co-Diagnostics. We're a molecular diagnostic company in Salt Lake City, Utah. Our mission statement is, at Co-Diagnostics, we are passionate about providing the most robust and innovative molecular tools for detection of infectious diseases, liquid biopsy for cancer screening, and agricultural applications, especially to those regions where pricing is paramount. Because we own our own platform, we're able to accomplish this faster and more economically, allowing for wider margins while still positioning Co-Diagnostics to be the worldwide low-cost leader of molecular diagnostic services. And that is pulled directly from our website. And we have a short, cheesy video for you to show you our lab. Let me know if you can see this.
Also, the audio should be working.
Okay.
But stop us, Deidre, if it's not.
So I think that.
What's that? Okay.
Yeah, I can Kyle, but I don't.
What's that?
The audio is working, but the visual is not.
Okay, I think we have to share the other screen.
Yeah, do a share.
How do we get to Teams here? Wait. This one? Okay, sorry guys. This one.
There it is.
Okay, now can you see it?
Yep.
Yes.
Oh, perfect.
Oh, and now your visual is working, but your audio is not.
Oh my
Okay.
gosh.
Whoops.
We got it to work before.
Yeah, did I not click? Oh, I did the same thing again. Wait. Whoops. I want to X that. Wait, this button. Sorry, guys. Where is? Oh, there it is. Okay.
All right, we got it. We know what we're doing.
Yeah. Okay, here.
Good.
Sounds great.
All right, so here we go. So that was just a little introduction to our lab. And you could see some of our Co-Dx box. That's our PCR machine that we use. All right, so a little bit about our history. Dr. Brent Satterfield founded Co-Diagnostics, and he also invented co-primers, which is one of our patented technologies that we'll talk about later. The CEO is Dwight Egan, and he brought a lot of the business into the science, so they have a really good partnership. And we have a global partner with CoSara. They're in India. We've been with them since 2019. And in the year 2020, we have grown a lot, especially in our Utah location. We have doubled the size of our lab. And as Kyle mentioned, they've grown from 2 lab techs to over 10 lab techs. I was the 4th one to get hired in February, and now we're looking to hire even more people. So we're growing rapidly. A lot of that's due to the pandemic, but we're also doing a lot of really cool research here. We also have purchased a lot of new robots, so they help us with extraction, and we have many different PCR machines, so lots of new technology and new science. Some of the products that we have are, well, the most popular ones are the COVID-19 diagnostic tests. The first one we came out with is our COVID-1, which is a single COVID gene target. That's the RDRP gene that we detect in that test. And the COVID-2, we just added a second COVID gene target. So we have RDRP and gene E in the COVID-2. And in both of those, COVID-1 and COVID-2, we also detect Rnasp, which is a human control, just to make sure that our tests work on the human sample. So when you run it, you'll see results for Rnasp, and then also if you have COVID, that'll show up. We also have ABC, which is influenza A, B, and COVID-19. So that multiplex will be able to differentiate between all 3 of those diseases. So it's very important to have during flu season to see people who had symptoms to figure out if they had the flu or if they had COVID-19. And our most recent COVID-19 diagnostic test is the direct saliva. So direct saliva is really neat. You just take saliva and add proteinase K to it, and that's what you do instead of doing a full extraction. So it's much faster, easier, and it has the 2 COVID gene targets. And I know some of you have heard about the variants out there currently. So our tests actually still work even with all the variants, because the 2 targets that we chose are very conserved, and the gene that mutated on the new variants are not at all the genes that we detect with our tests. We also have vector control products and RUO tests. So I just wanted to show you that we do other things besides COVID, but today we're just going to focus on the COVID-19 diagnostic tests.
So what you're seeing here is the COVID-001, or the first version of the COVID test that we developed. There are 3 components that come in the test kit that we sell. There is a master mix, and that's basically the test really is the master mix. Then you have a positive control, which just functions to make sure that your test is working correctly. And then the nuclease-free water acts as your negative control to make sure you're not getting false positives. So 3 components. You have master mix in every sample that you'll test, and then you'll run 1 negative and 1 positive control along with any of the patient samples. So that's the extent of what comes in a COVID-19 test kit for us. And then our test is a PCR-based test, and I'm not sure who is familiar with PCR in particular, but I thought I would just at least introduce it to make sure everyone kind of understands what we're talking about here. So PCR stands for polymerase chain reaction. Carrie Mullis won the Nobel Prize for PCR in 1993. PCR has really proven to be kind of the most powerful research tool that scientists have when it comes to genetic research. It's a method to reproduce a sequence of genetic material in an exponential fashion, so you can make millions to billions of copies of a specific chunk of nucleic acid. And this has many, many different research and diagnostic applications. Our test is a real-time RT-PCR test. So what that means, to break it down sort of, is real-time means that we can track the progress of the reaction in real time. So you can actually observe the progress of your reaction as it's working. And we'll talk about how we do that in just a minute. The RT-PCR portion, the RT stands for reverse transcriptase, and that is an enzyme that converts RNA into DNA. So COVID-19 is an RNA-based virus, and there's many other viruses out there that are RNA-based. So this isn't very uncommon. So our test is real-time RT-PCR, meaning we have an RNA template, or COVID-19, that we convert into DNA, and we can track that progress in real time. Again, to sort of zoom in on PCR, this is kind of the way that PCR works. You cycle from a high temperature to a low temperature to a medium temperature, and then back up to the high temperature. And this cycles for 40 to 50 different cycles. That first stage, you heat it up, is your denaturing stage. So you'll take your double-stranded DNA and make sure that it's into 2 separate linearized strands. You'll drop the temperature down. Your primers will swoop in and sit down on that template. And then the temperature will raise back up a little bit, and that's your extension stage, where the polymerase will basically generate a brand new copy of that chunk of DNA. And this just happens over and over and over, and you'll get an exponential amplification of that target sequence if it is present in your sample. And so this is kind of how PCR is traditionally done. And so I'm going to call it a normal primer here on the left. This is just a chunk of DNA, and this is what's called a primer. That chunk will sit down on your template. The polymerase will come in and it'll extend down in the bottom left there, and you'll have a brand new complementary strand of DNA. And this is the traditional sort of PCR that many other diagnostic tests will use. But in the PCR diagnostic world, the biggest issue that people ever see is something called primer dimers. Primer dimers is basically, it's a way that you will generate false positives. So false positives are never good, right? So you really want to avoid primer dimers at any cost. And this is always the pitfall of any PCR-based test. It's basically primer 1 sits down, has a little bit of overlap with primer 2, and then you get primers amplifying on primers. So they're not actually amplifying a template, they're amplifying off themselves. And so that's how you generate the false positives with these traditional primers. But Co-Diagnostics was founded based on a new technology called co-primers. And so this is kind of Co-Diagnostics' big thing. This is the patented technology that we use to develop all of our tests. It's basically taking the normal primer and splitting it in half. So we take that full primer sequence and we split it in half so that we have one short primer sequence up in the top right, that little blue chunk, and then on the far left you see the capture sequence. So both of those together would be the size of a normal primer, but we separate them with an inert linker sequence that is not genetic material. It's just like a carbon chain. And so effectively what we do by splitting it in half and separating them is that we say each individual chunk is too small to actually produce an effective amplification. So if one of them has a single mismatch on either side, you're not going to see anything happen. But as long as both of them are able to sit down on that template, then you will see a reaction occur. And so this has a lot of advantages that we'll talk about in just a second. But the big thing is that on that 5' end of that purple chunk, we can label it with a fluorophore. So as the blue primer sequence amplifies, the polymerase will come through, chew up that purple sequence, and release the fluorophore. And every time that happens, it emits another little burst of light. And the more light that gets emitted, the more signal we receive, and that's how we achieve the real-time portion of our test, where we can observe the reaction happening at real time, because you'll basically see the development of more light over time. So co-primers have the advantage of preventing primer dimers, is really the big takeaway for co-primers. So they were invented as a way to reduce primer dimers and effectively eliminate the false positives from happening. And so that's really the major advantage of our technology here, is we don't get false positives coming up when the co-primers are working correctly. Additionally, they have an increased specificity. So like I mentioned, if there's a single base pair that's missing on either one of those chunks, that will be enough to keep the reaction from happening. So we have some applications that are really useful for this, in particular in cancer research, where a single base pair could result in a horrible cancer of some sort. So co-primers have the advantage of being able to detect those kind of single base pair changes. The way that we go about developing a diagnostic test, sort of our workflow in general, is first off, identify a disease of interest. The big one, obviously, that everyone is familiar with right now is COVID-19 or SARS-CoV-2. So that, we identified that back in December of 2019, I guess. And we realized, this is an emerging disease. We need to get a test ready for this. So as soon as the full genetic sequence was released, we combed through it and we ran it against all different other similar organisms that we could find that were available. We made sure that the sequence that we were interested in identifying had no overlap with other organisms. It was highly conserved with any of the known COVID-19 strains. And it was going to be a sequence that would be non-mutating. So like Karlee mentioned, those new variants that are coming around are mutating in certain regions, but we were able to identify a good region that we thought would not be mutating, and luckily we were right. So after that part, we know what our target is, so we can design co-primers to target that sequence there, which we did. We ordered those co-primers and we tested them, selected the pairs that were working the best. And from there, then we entered the validation studies. So this is basically just a big checklist of things to understand about how your test performs. So you want to make sure that it's not going to cross-react with other organisms. You want to see what your limit of detection is, see basically how few copies can you detect. You want to make sure you're not getting false positives. You want to see how reproducible it is, etc. So that's kind of the workflow for developing a diagnostic test.
Yeah, so the first thing you do if you want to test someone for COVID is you have to first collect a sample from the patient. There are a lot of different sample matrices. I'm sure most of you are familiar with the NP swab, so those are a little swab that goes in your nose. It's very comfortable. Then there's the saliva method, so you can just spit into a tube and process your saliva, and there's also sputum. So those are the three main ones. there are a lot of different extraction methods, a lot of different brands out there, but they all have the same general method where you collect your sample and you lyse your cell, and then you have to clean your sample that's been lysed. So in the middle there's a diagram of a column. So the RNA will bind to the column, and then you can wash out the extra proteins and lipids with different wash buffers. And then when you want to get that RNA off the column, you just elute it with usually water or water with some PK in it, and it will become unstuck from that column, and then you have a very concentrated sample of RNA. There's also magnetic beads that do the same thing. It's very cool to see all the little magnets stick to the side of the tube, and then you can remove all the wash from it. It's essentially the exact same, just with small magnets in the solution and then putting it onto a magnetic tray to extract. We also have direct saliva that I mentioned before. So that is a new method of extraction that one of the doctors here invented. Lu Ming invented direct saliva. So that's just saliva with proteinase K, and you heat it and it's ready to plate right away. Here are a few extraction machines that we've purchased this year. We used to have just a single QIAcube, which is the machine on the right, where there's two of them. That broke on us during some of our FDA studies, so we had to purchase another one. So now we have one that works really well, and it's really convenient. You can do 12 samples at a time and just load the buffers into the machine, and it saves us a lot of time. You just like set it to whatever extraction method you want to use, and it does it for you. And same sort of thing with the Maxwell. I think the Maxwell is actually even easier to set up with the buffers. I haven't actually used the Maxwell, but I have used the QIAcube a lot. And the Rt Pcr is amazing. It has 8 or 9, I think it's 8 plates that you can place on it at a time, and each plate has 96 wells in it. So you can do a lot of patient sample extractions at a time. Very cool machines, and they're fun to have now. So after you process your sample, then you can move on to plating. I have pictures here of our Co-Dx box and our plates that have our wells in them. So with the black top on the caps, those are the plates that we use. So these are 48 well plates, and you can run 46 patient samples in a single plate with these. You do need to have one NTC and one PC, so that's your non-template control and your positive control. Those will tell you what a regular negative result should look like. So that non-template control should have no amplification, it'll be a flat line, and your positive control will have a nice curve to it because it should be amplifying. And if you don't see your PC control amplifying, then you know something is wrong. And additionally, you can run a known negative sample, and that will tell you if your internal positive control is working, so that IPC. So the internal positive control is an additional gene target that you would find in, let's say, a saliva sample. So the Rnasp gene is a common human gene, and if you see that amplifying in your test, then you know that your extraction was successful. And if you don't see it amplifying, there probably was some sort of error that happened during extraction, or someone gave a poor sample, like they had, I don't know, just eaten or something, and it inhibited the extraction. So those are the general rules to plating. A PCR run lasts about an hour, and I have some results over here. So the first thing you look at is your Rnasp Target. So you see the bright yellow curves. So that means that all the samples, their IPCs have amplified. So that means all the samples were extracted properly and the patient gave a good sample. So that looks good. If it didn't amplify, then you'll probably want to collect a new sample and do a new extraction. And then you can look at your COVID target. So in this case, there's only a single COVID-19 target. So this is the RDRP gene amplifying here. So there aren't as many amplification curves. So some of these samples were positive. And the negative ones, there's a purple line that you probably can't see, but those are completely flat. So those would be your negative patient results. And there's something called a CQ value. So PCR runs for about 50 cycles. So every heating and cooling step is a cycle. And the positive control comes up somewhere between 20 and 24 cycles. And the later the CQ value is, the more diluted of a viral load is in your sample. So a patient that's positive with COVID, you could potentially see something pop up before the positive control, but generally it's before 37 cycles. And you probably have all heard of some COVID-19 tests causing false positives or false negatives. So a few things that can cause that are nonspecific amplification. Those would be your false positives. So like Kyle mentioned before, primer dimers are a huge problem with PCR testing. So that's where the primers are binding to each other and amplifying. There can also be a nonspecific amplification from degradation from your master mix. So if a scientist or doctor mishandles the master mix, it can cause nonspecific amplification. And you can tell it's nonspecific amplification because the fluorescence is really low compared to your positive control, and the CQ value is usually much later than your positive control. So after 40, you'll start to see CQ values pop up for those. So ways to mitigate that are just to not do too many freeze thaws on your master mix. Don't leave it at room temp. Always plate on an ice block. Do whatever you can to prevent your master mix from degrading, because that's when you'll start to see problems. And for false negatives, we've seen a couple sensitivity problems with other tests. So if a test isn't sensitive enough, it won't be able to detect a really low viral load. We actually had a customer complain that our test was producing false positives, and we found out that our tests were actually just more sensitive than the ones they were comparing it to. So we were the only ones able to detect a true positive, but it did look like a false positive. So sensitivity can play a big role in false negatives, and that's just the nature of having so many different PCR tests available, is that the sensitivity will vary from test to test and even with different extraction methods.
Yeah, so one of the big things that you want to evaluate any COVID-19 test on is the sensitivity, or the limit of detection. So how few copies can you detect? Any test's limit of detection is defined as the concentration of template that you can detect 95% of the time. So like Karlee mentioned, we've had customers who are comparing 5 or 10 different tests to each other to see which one do they want to buy. So they'll have our test in the panel, and they'll see that all 9 of these tests will come up negative for a given sample, but our test will come up positive, and it has a really late value. And so they'll say, hey, your guys' test is giving us false positives. But when you compare our sensitivity to those other tests, what we see is just that our sensitivity is better than those other ones. And so this table kind of shows you our limit of detection studies. So in the far left, you can see copies per reaction. So this is where we made known dilutions of positive template, basically, and diluted it down, starting at 50,000 copies in the reaction, going all the way down to a single copy in the reaction. And you can see the detection rate, second from the right, that column. We get 100% all the way down through 10 copies per reaction. The next dilution is the 7 copies per reaction, and we have about 98% detection rate. So this COVID test for us has a limit of detection of about 6.8 copies per reaction. That's the 95% detection rate. So that's a pretty low number compared to a lot of other tests out there. And so this is kind of a normal sort of study that you'll see for limit of detections evaluations there. Additionally, like Karlee mentioned about the false positives, you can see those 2 bottom rows, the NTC, or the no template control, negative control basically. And there's also the one called negative. And the negative was known negative samples that were donated. And so you can see that there are 0 detected out of those, 43 samples there. So our test has a sensitivity down to about 7 copies per reaction and is not producing false positives. So that's kind of a basic evaluation of the sensitivity and reproducibility of the test there. So Karlee did already mention some of the new or upcoming products that we were looking to develop. One of the big ones right now is still in COVID-19. It's the direct saliva test. And so she kind of went over how that one works, having just a very quick throughput, very efficient throughput. Some other sort of upcoming or ongoing development efforts that we have. One of the big ones is MTB, or Mycobacterium tuberculosis. Tuberculosis, before COVID-19, this really has always been the #1, the disease responsible for the highest number of deaths per day worldwide is MTB. So this is a disease that is horrible and is very impactful in a lot of developing regions where there's not as much money being thrown to research efforts. And Co-Diagnostics, as part of our mission statement, is to bring low-cost, efficient, high-quality tests to developing regions. So MTB is a big focus for us. Another research thrust for us is the H's, the HIV, HBV, HCV. And so that's another ongoing effort. And I mentioned previously about cancer screening. So liquid biopsy cancer screening is another really big interest of ours. And coprimers have a very unique property in their sensitivity to single base pair mutations. So this is another big effort for us coming up here.
Yeah, so just a recap of this year during a pandemic while working at a diagnostic company. So we started developing the COVID-19 test in mid-December, and then a little bit later, I think it was early March, it was the quarantine, and I got hired maybe 2 or 3 weeks before the big quarantine. And we're all working in the lab because lab work can't be done from home. And in March, they declared it an official pandemic. In April, we got our first FDA approval on the COVID-001 test kit. And since then, everything has exploded. Our lab space has doubled, and our offices have also maybe doubled or tripled in size. And now we have our vaccine out. Well, not our vaccine, but the vaccine is out in 2020, so that has been amazing. and we have produced over 13 million tests in 2020. So it has been an extremely crazy and busy year, but we have all made it this far, and I am really happy to have experienced working in a diagnostic lab during a pandemic. It is once in a lifetime.
So yeah, at this point we're basically done with the presentation we had prepared, and we just wanted to throw out there, we are a company that is kind of growing and expanding. So if anyone is interested in any sort of opportunities after you guys are graduated and everything, we just have our R&D director here quoted as just saying, always looking for promising talent. So, if anyone's interested, contact Karlee or myself, and we'll be happy to direct you to the right avenues. But other than that, if there's any questions, we'll be happy to field those now as well.
Okay, so no questions came in through the chat. But I see a hand has popped up, and it looks like it is Dr. Yang.
Dr. Yang, I believe you may be muted.
Yes.
Sal, you're muted.
We still can't hear.
You're muted, Sal. We cannot hear you. Unmute yourself.
Looks like he's trying to figure it out. Let's see. Well, maybe you can type it in the comments. If there's anybody else who has a question, we can jump to them. Students?
I got a question.
Yeah.
So you have all those different tests. How far does your company distribute those? Like, is it worldwide, or is it just in the Americas, or how far is it distributed?
It's definitely worldwide. Yeah. The COVID-19 in particular, a lot of it is in America, but we definitely are distributing worldwide for COVID-19. We have a few big distributors in Italy or in Georgia, in some different African regions. So there's a lot of different areas that the tests get sent to. There's one really big Saudi Arabia distributor. So yeah, it's all over.
Mm-hmm.
Some places are harder to ship to than others, but yeah, we can get it there.
Yeah, the shipping world of this is a pain.
That's a whole nother world.
Well, I have a question for you. You have these master strains that you do periodic testing of your test kits. Do you multiply and keep track of your own strain, or do you get a new supply from a master library that, I don't know, maybe the CDC controls or whatnot?
Yeah, so we are not a lab that's set up to maintain our own live strains. We don't have that BSL certification ourselves. So all of the strains that we receive are inactivated by chemical means or heat inactivated, some method. But those are all received from another lab. And so most of that's done during the validation studies. We'll just get our hands on as many different ones that we could possibly find. And then, once that's all taken care of, then, we're not maintaining those ourselves. But, as new strains will pop up, in particular the new variants that are arising, then we'll test those, as needed.
Okay.
Ethan has his hand up.
Go ahead, Ethan.
Yeah, so when you were talking about how he kind of identified like the COVID gene to target on your test and stuff, how, like, what was your process in doing that? Did you, when you found like that certain gene, did you like use, like, what type of test did you use, or how did you kind of decide this one probably will not mutate or something like that?
So we actually have a really great guy, Mason, who works here, who selected the genes, and correct me if I'm wrong, but he used machine learning to select the genes, and then we did further testing in the lab to confirm that they worked well. So I don't know exactly how machine learning works, but that was the initial selection of the primers for the gene and selecting of the target genes.
Yeah, and so additionally too, when the sequence was released, you can kind of compare some of the sequences to other known sequences where you know this particular gene is a structural gene, or this is the spike protein gene. So we selected genes that we knew were structural and that were not going to be mutating. So just parts of the viral envelope that would not be changing as much. So initially, those were kind of the targets where, you know, those aspects of the virus weren't going to be changing a bunch. And then from there, then he could take those targets and put them into that machine learning program that is another sort of like patented part of the company. This was developed by Co-Diagnostics. So he was able to use those targets, put them into his system, and then it sort of output the ideal co-primer targets, basically.
Gotcha. So something kind of similar to like protein domains?
Yeah, absolutely. Yeah. Parts of the protein that wouldn't be surface exposed as much, where they're more structural. So yeah, like the spike protein, there's highly variable portions of that spike protein, but there's other structural components that wouldn't change as much. So you would never want to target that variable part unless you're trying to detect different strains. You know, so maybe your purpose was to detect those variations, but our purpose is to find one that's robust and would not be affected by any mutations. Yeah.
Awesome. Thank you.
Yeah.
I have a question. So in that graphic you had put up of the co-primer, and there's the capture primer end. After the replication starts, it looks like the capture primer end is like destroyed when the replication hits it. Has that caused any issues, or like, does that mean anything? What did that change compared to just the normal primer test?
So that's actually what releases the fluorophore, and that's how you get the emission of light so it can be detected.
Yeah, so that's working as designed.
Yeah, exactly.
It's that, that tail that's left behind?
So that capture sequence gets destroyed, like you saw, and by it getting destroyed is what produces the signal. So that is desirable, really. So that's kind of designed in the way on purpose so that that portion will be destroyed. And every time that is destroyed, it releases another burst of light, basically. And that's how we track the reaction.
Okay. Thanks.
Okay, so we're closing on our last few minutes here. Technically, we've gone over for the class time, which always happens. I'm going to close with one last question. You guys both come from kind of a biochem, cellular biology background. What is the one skill set you would recommend for students who would be looking to get into a field of industry that you work in?
Work on your pipetting.
Yeah, definitely as far as like lab skills, definitely pipetting. Another big one I would say is being comfortable, almost, I kind of want to say learning how to learn, I guess, because you really can't anticipate every job that you might apply to. But as long as you're someone who is capable of learning new techniques, willing to read on whatever techniques you might be learning, you know, just being willing to learn. And so I was always told, learn how to learn. So that was kind of one of my biggest things from school, and I think that was a pretty valuable skill coming into industry.
Excellent. Well, thank you so much. Please everybody join me in thanking Karlee and Kyle for their awesome talk.
Thank you so much.
Thank you.
So both of them do have LinkedIn pages. You guys can connect with them. They were sent out in the original announcement for this seminar. And we have another seminar right away next week, Friday. So keep your eyes on your email for that. Thank you everybody for attending.
Thanks everybody.
Thanks for having us.